Hello everyone, myself B. Anita from 2nd MSc Microbiology, Sacred Heart College, Tirupattur. And today we are going to discuss about lactic acid production. Lactic acid. Lactic acid is a type of organic acid which was firstly produced from the fermentation process in 1880. The chemical formula for lactic acid is C3H8NO. It is also known as milk acid. At first, the lactic acid was extracted from sour milk by Swedish chemist named Carl William Siegel in 1780. When lactose undergoes fermentation, lactose is a milk sugar. When it undergoes fermentation, lactic acid is produced. And this is the chemical structure of lactic acid, which occurs in two forms. One is D form and another one is L form. It has a functional group known as COOH, an hydroxyl group OH, and a central carbon atom having an alcohol carbon group. Microorganisms involved in the production of lactic acid are both homofermenters and heterofermenters. Homofermenters is nothing but when the fermentation takes place, a single end product is produced, that is typically lactic acid. And heterofermenters is nothing but when the fermentation takes place, one or more different products is produced, typically alcohols and lactic acid. The homofermenters examples are Lactobacillus bulgaricus, Lactobacillus plantarum, Lactobacillus delbrueckii, Streptococcus lactis, and Pediococcus species. And the heterofermenters are Leuconostoc mesenteroids. Biosynthesis of lactic acid. The glucose is a primary source for producing lactic acid. The glucose is converted into glyceraldehyde 3-phosphate. When the phosphate enters the reducing power, which is changes into NADH. The glyceraldehyde 3-phosphate, with the help of the enzyme glyceraldehyde phosphate dehydrogenate, it converts into 1,3 diphosphate glycerate. Then the glycolysis cycle takes place and it is converted into pyruvate. The pyruvate is then reduced to produced as lactate, which is with the help of the enzyme lactate dehydrogenase. Fermentation process either takes place in batch fermentation or fed batch fermentation, and the pH is maintained by 5.5 to 6.5 by using calcium carbonate, and the period duration of fermentation is 5 to 10 days. 2 to 13% of glucose is used in the medium when the pH is maintained at 6.3 to 6.5 with a continuous neutralization of acid. The optimum temperature is 45 to 50 degrees Celsius. For Lactobacillus delbrueckii and Lactobacillus bulgaricus, the pH should be maintained at 45 to 50 degrees Celsius. For Lactobacillus casei and Lactobacillus pentosus, it requires about 30 degrees Celsius of temperature. Aeration and agitation is required and the yield gives about 93 to 95% of yield when glucose is given. When lactic acid can also be produced by fungi, rhizophus or rhizae, by both batch fermentation and continuous method. The raw materials includes carbon source, nitrogen source and growth factors. The carbon source such as glucose, maltose, sucrose or lactose, and crude substrates such as maize starch, potato starch, rice starch, whey, molasses, etc. And nitrogen source, ammonium salt is one of the major nitrogen source used here. The ammonium salt is used as ammonium hydrogen phosphate used in the manufacture of lactic acids. Growth factors. Since the lactobacillus needed complex nutrients, vitamin B complex is used as a growth factor. Harvest and recovery. The fermentation broth is filtered by using porcelain filters. Porcelain filters is nothing but it consists of a tiny spores that allows only the fluid to pass through while holding back the microorganisms. This filtrate is acidified with HCl to regenerate lactic acid and then washed. The washed filtrate is treated with activated carbon to remove the organic impurities, and then the evaporation steps is taken to increase the percentage of lactic acid about 50 to 60 percentage, and then it is treated with ferrocyanide to remove the heavy metals. It is finally purified by passing through ion exchange resins. Uses of lactic acid. It is mainly utilized in food industry and medicine. It is used to manufacture polymers, and it also helps in preventing the coagulation of milk proteins. Utilized in textile and leather tanning industries. It is also used to recover traumatic brain injuries. It is also used as a blood coagulant and calcium source for diet. Thank you for listening.